Hi everyone, I've made sure that you can hear me this time, so I'm using my headset. Alright, this week we're going to be doing some activities about enzyme activity. Now what we're going to learn is we're going to describe relationships between structure and function, which is very, very important as far as enzymes go. Relate structure and function to active sites, modes of inhibition, and optimal conditions for enzymatic activity. We'll talk about all three of those things shortly. Hypothesize and test how inhibitors and changes in temperature and pH affect enzyme reaction rates, so how fast or slow something can happen. And describe how some enzymatic reaction rates can be measured by color changes and gas liberation as products are formed. So we're going to be doing all kinds of things. So enzymes are biocatalysts. So they accelerate reactions or start reactions, and they work in biological settings. So in the body, in humans and almost all organisms, chemical reactions in the cell are not spontaneous, and that is a good thing. It's a good thing they're not spontaneous, because if they were, things would be quite chaotic and uncontrolled. So we have these special proteins called enzymes, and they help control reactions. They let it happen in a more regulated manner. So what the enzyme does, it binds to a substrate to form what's called an enzyme substrate complex. So if an enzyme, I like to make my enzymes look like Pac-Man because it's easy. We have an enzyme here, and its substrate fits right into what we have, the active site. So the active site is where the substrate binds, and when these are bound, so let's make a Pac-Man here, we have this enzyme substrate complex. Now what happens here is that usually the enzyme changes shape in some way. So let's say it gives Pac-Man, I don't know, a tail, just as an example. It changed shape in some way when this substrate binded to the enzyme. Bound to the enzyme. So the enzyme substrate complex creates what we call a transition state for the substrate, which means it changes a little bit. So it's not stable in its normal configuration, and it's not completely broken apart or added things together or anything. It just is slightly changed. So what happens is that the bonds are stressed. So the example they use in the next picture is that they have a sugar, looks like a disaccharide, and the goal is to break it. So when it goes into its enzyme substrate complex, it stresses the bonds. So we can demonstrate that by showing a bond being not completely straight between these two sugars here. So it becomes more reactive. This stressed bond is more reactive than the stable covalent bond here. So what happens is that now the energy needed to break this bond is lower than it was in its original state. So the energy needed to form this transition state, to go from here to there, to change this bond shape, is called the energy of activation. So the amount of energy needed, cross that out, to go into the state. So the enzyme lowers this energy. So when the enzyme binds, it means that it requires less energy in order for the reaction to occur. So a product is formed after the reaction, and the enzyme returns to its unbound shape. It returns to its original shape. That's the important thing, and it's ready to accept another substrate. So the enzyme is a reusable molecule. It binds a substrate, it changes shape slightly, it releases the product, and then it goes back to its original shape. So here's an image from your text about the enzyme and the enzyme substrate complex. Now, I don't know what enzyme this is, though some sort of enzyme. And notice that the active site kind of closes around the substrate in this case to form this enzyme substrate complex. And it also becomes a little bit more compacted. Notice this area here. And it's also a little bit shorter in this area. So it changes shape. And all of these things I think are representing poly or peptides, so the amino acid chains here. So here's an image from your text as well. This is figure 11.1. And this talks about energy of activation. So activation energy is like a hill. It's like a hill that you can climb, or as I like to use the analogy, ride your bike up, because I like riding bikes. So you have this huge amount of energy, not necessarily huge really, but an amount of energy that peaks right up here. This is the activation energy. And you have to have enough energy to get all the way up this hill and over in order for this product to form. Even if it's combining two things or breaking apart things, everything has its activation energy for a reaction. So with an enzyme, so it creates that, remember the transition state for the substrate with this stress bond. This is now created by the enzyme, and so now you have much less energy in order to complete the reaction. You don't have nearly as high of a hill to climb as you do with the enzyme. Here's a nice image as well. This is found on page 114, figure 11.3. So this shows a disaccharide, I assume. It's probably, let's say, oh, it's sucrose. Yep, disaccharide. So glucose and fructose, here's glucose, it's a 6-carbon, and fructose, a 5-carbon sugar. And they're bound here in the middle. Now when they bind to sucrase, sucrase is the enzyme. So enzymes end in ase. That's an easy way to remember enzymes. So we have, say, DNase, RNase, helicase, polymerase. So many different aces. All of these are enzymes. So this sucrose binds to the active site. So there's a spot precisely for the glucose and precisely for the fructose. Now active sites are very, very specific. They will often only accept one type of substrate. Very specific. So this enzyme and the substrate will bind, and we create this stressed bond, which is currently lowering the activation energy. The energy needed to overcome that hill and create the product. So in a hydrolysis reaction, so hydrolysis means hydrolysis, so breaking by water. We add water here to the bond to create an O, I think it's an O and an OH in some way or shape or form, and we release this as glucose and fructose. So the steps: substrate binds, bond is stressed to create the transition state, product is formed, and now the enzyme is ready to receive another substrate. So here is just a flow chart of that exact thing. Enzyme plus substrate means enzyme substrate complex. So after the energy has been overcome, then you have the enzyme and the product as separate things, enzyme released, and the enzyme resets itself. So enzymes are proteins, meaning they're long chains of amino acids, and they're highly folded into complex shapes. And they have to have, remember, that specific active site, like my weird Pac-Man enzyme here. Pac-Man has a tooth. So it has a specific shape, and this active site must be just right to fit whatever its substrate is. So remember this important point right here. An enzyme's function depends upon its shape and catalyzes a specific reaction. So active site usually accepts only one type of substrate. So that means a specific reaction. On the previous slide, we had sucrase, and the only thing sucrase can do is break down sucrose. That's all it can do. Amylase, for example, the only thing it can do is break down amylose. Or DNA polymerase, all it can do is create DNA polymers. That's it. Specific reaction. So because an enzyme's function depends on its structure, any structural changes mean that the function changes. That makes sense, right? It changes the way the enzyme can act because it depends on that structure so much. If an enzyme denatures, that means it straightens out. It loses its 3D structure. So if we have this complicated enzyme, here's its polypeptide chain. When it denatures, you end up with something that is a different shape, and often it's just a chain of amino acids now. And in high heat or low heat, or high pH or low pH, so extreme environments, it can do this. It can denature. So for example, when you cook eggs, if you cook eggs, they will be runny until you put them in the hot pan. And then the white of the egg starts to turn white instead of clear. And that's happening because the proteins, the albumin, which is just a water-soluble type of protein found in eggs, when it denatures, it forms basically a solid instead of a kind of gel. So if you were to try to use any of the enzymes for their purpose while they're denatured, you probably wouldn't get any kind of reaction because this shape, as ridiculous as that might look, no longer is available. There's no active site anymore. There's just a chain of amino acids. So enzymes all have optimal conditions in which they work. So think about this in terms of the human body. And there's a couple of questions, at least one in your lab manual that you'll have to answer that is a thinker. It makes you kind of consider this. So an organism's environment, if an organism has enzymes, which it does because they catalyze the reactions, then they have a specific environment that they work best in. So usually it's a specific heat or a pH that they work best in. So generally these two things are the biggest factors. So heat or pH, one of those things change and you could change the total functionality of your enzyme. So in humans, human enzymes work depending on where they are. So we have enzymes in our mouth, in our saliva, that break down food. Now the saliva in your mouth should be generally pretty neutral. If it's more acidic, you might have some bacteria in there. But in the mouth, the pH is generally pretty neutral, but there are also enzymes in the stomach. The stomach has a really, really low pH. It's super acidic. And those enzymes can still work though, because that is their optimal condition in which they exist. So here is something, talks about temperature and pH of the reaction. So the rate of reaction, it shows in the arrows on the left pointing up. So the higher the rate of reaction, the faster it's going. So in heat, we generally have a pretty linear slope that gets faster and faster and faster until we reach its optimal temperature for human enzymes, for examples. And so the optimal temperature it's pretty close to body temperature. So 37 degrees Celsius is body temperature. And so once it reaches this threshold, however, the functionality is no longer. We can't use it too much after that heat threshold because it will denature, it will lose its shape and its ability to bind to the substrate. So for example, hot springs prokaryotes, some of the most ancient organisms, they have, look at this working temperature. This is very hot, 70 degrees Celsius. So if 40 degrees Celsius is about 100 degrees Fahrenheit, then 70 is almost 200. It's probably closer to about 175, 180 degrees Fahrenheit. That's extremely, extremely hot. That is nearing boiling at 212. So these hot springs prokaryotes, they have specific enzymes that have temperatures that are optimum at around 70 or 70, I don't know, 72, 73 degrees Celsius. Extremely hot temperatures. And so even at our body temperature, those could not work at all. There is no zero reactivity there. So enzymes are specific to their organisms based on their temperature and also pH. So pepsin is a protein in the human body, and I'm not sure where pepsin occurs. I'll look that up in a second and say it on the next slide so I don't waste any time. So pepsin has an optimal pH range of about 2.5, and I suspect because that this is a low acidic pH that this is one of the ones found in the stomach. However, trypsin operates at approximately a 6.5 pH. So this is very, very slightly acidic here. And so I suspect that this is not found in the stomach, but I don't honestly know where trypsin is found. So I'll get back to that in the next slide. Temperature definitely has an effect on enzyme activity. Remember the previous slide where we had this chart where we have a nice gentle slope upwards as heat is increased, and then we have a sudden drop off. So heat increases this way and the rate of reaction increases this way. So we increase the random movement of molecules. So remember Brownian movement, we did that a few weeks ago where you saw these little particles just kind of vibrating underneath the microscope. They were Carmine powder and they were so small, they weren't very big so that you could see them moving. So the random movement of molecules means that the enzyme is more likely to bump into the substrate and vice versa. They don't go seeking each other, they just happen to bump into each other. It's like meeting somebody you like in the grocery store kind of thing. You don't necessarily mean to meet in the grocery store, but you see them in Hy-Vee anyway. So as this heat increases, the rate increases until we reach this high point here. And at this high point is usually when the enzyme is denatured, meaning it is simply too hot to function. Being a Texan, I totally know what that's like. So the protein is the enzyme that is a protein is denatured and the active site is no longer able to fit the substrate. So imagine if my Pac-Man here suddenly had a round mouth and looked really silly and could not fit that triangular substrate anymore. That would not work. So it changes shape and can no longer fit. So in catechol oxidase is the enzyme you're going to be using for the next experiment. And it is a plant enzyme found in potatoes, so I'll be making more potato slurry. And the catechol oxidase is going to break down catechol to benzoquinone, which is the brown in bruised fruit. So anytime you bruise a banana or an apple or whatever it is, that brown is that compound called benzoquinone. So what we're going to see is a brown color develop as more catechol is broken down. The catechol concentration will decrease, the benzoquinone will increase, and we will see a brown color. So I looked up pepsin and trypsin, and pepsin indeed is found in the stomach with that ideal working pH of around 2.5, which is very acidic. And we have trypsin found in the small intestine and with a working pH of around 6.5, much less acidic than the stomach. So in procedure 11.1, you're going to be determining the effective temperature on catechol oxidase activity. Catechol oxidase is an enzyme. So you're going to start with a substrate called catechol and add the catechol oxidase enzyme. I think, let's see, that is found in, I believe it's potato extract. I'm going to be making some potato extract for that, grinding up more disgusting potatoes. So that is our enzyme that's going to break down this catechol to create benzoquinone, which is a colored product. It has a specific color to it. Now what you're going to do as part of the experiment, you're going to propose hypotheses about what temperature will do to the catechol oxidase. I will have already prepared water baths at the specific temperatures that it asks for. So it says 22 degrees Celsius, which is room temperature, 4 degrees, which is fridge, 40 and 80. So I'll have a 40 degree Celsius and 80 degree Celsius water bath ready to go. And so I will assign you all a group letter. So in group A, for example, you'll have group A tube 1 ready to go in the water bath. Group B will have tube 1, group C will have tube 1, etc., etc. So that I only have to prepare 2 water baths at 40 and 80, and you don't have to prepare your own, but you'll be able to recognize your tube when it's time to look at them. So for part 9 of this procedure, we will not be doing any further quantification by the spectrophotometer. And I'll give you a rundown on how to do that because you're going to be using it in procedure 3. Very, very, very important, catechol is toxic. It is not healthy for organic beings, which includes us. I will have a specific waste container. I will label it as catechol waste, and it will be in a very obvious place, probably over in the sink where I usually put all the stuff for the activities, so that that will be disposed of. Now you must wash your test tubes extremely, extremely well as well. And we're going to start this experiment first thing, before we even go over anything. I want you to start this because the lab manual prep sheet said it could take up to 2 hours to do this. I don't want to stay the whole 2 hours if we don't have to, so let's get it started ASAP. So the hypotheses you're going to create are going to be the null and alternative, that's what we call this. So the null hypothesis in any sort of statistical analysis or science means that the thing that you were testing has no effect, and that's what you're basing this on. So the null hypothesis states that temperature has no effect on catechol oxidase activity. And your alternative hypothesis would be, okay, well, which temperature would have the best effect if it did have an effect? So I want to see that in your lab notebook. I will be looking for it. When you are finished, make sure to clean your work area very carefully because there will be this catechol. I'm going to make sure to pick up some specific cleaner for these tables because first of all they're disgusting, and second of all because we need to make sure that we don't hurt anybody else after our work. Table 11.2 is found on page 116, and that will be part of your grade on the lab notebook to do that. Also answer question 1, and I will have graph paper available for you to paste in your notebook if you so desire. So we talked about temperature affecting enzymes. Well, pH affects enzymes in similar ways. However, the curve is a little bit different. So recall that heat was more like a slope with a drop off. It's more like a cliff here. Whereas the pH is more of a gentle parabola. So we have an optimum working pH, but it can still work some in the surrounding pHs for the most part. So in acidic environments where there are high concentrations of H plus ions, or in bases that have high concentrations of OH ions, what these charged particles do, if you got lots of H plus, for example, in an acidic solution, any negatively charged side chain, so I don't know of any examples, I don't have the amino acids memorized, but anything that happens to have, we're going to do X negative, these hydrogens will bind to them. And when that happens, it actually causes the enzyme to change shape somewhat. Because now there's no longer this negative charge that might have interacted with something before. So we have the amino acid general shape, or the general way that the main chain of the amino acid is. It's a nitrogen, a carbon, and a carbon. We have a peptide bond here. I'm going to put PB for peptide bond, PB for peptide bond. Here we have an O, we have an H, we have an H, and we have this side chain, which could be any one of 20 or more different things, depending on the amino acid. Let's finish out our Lewis dot structure here with those free electrons. So if it's a charged side chain, or even one that happens to be attracted to H plus or OH minus, then we will have some interactions that might have negative consequences on the shape of the enzyme. So they change the active site. So imagine, you remember my previous drawing with Pac-Man having now a silly round mouth and can't fit that substrate anymore? Same situation. It changes it enough sometimes. And the optimal pH depends on the environment it's in. So the figure 11.4 on page 115, it showed that the optimal pH in the for trypsin, which is found in the small intestine, has the optimum pH of about 6.5, whereas pepsin in the stomach has optimal pH of 2.5. So in the next activity, you're going to be observing the effect of pH on catalase activity. So catalase is another type of enzyme. So catalase breaks down hydrogen peroxide, which is H2O2. Hydrogen peroxide is highly toxic to cells. So we want to get rid of that as much as possible. So the formula is 2 hydrogen peroxides being reacted with catalase, which of course is the enzyme because it's got that ACE ending, will produce 2 waters and 1 oxygen gas. So in a ratio of 2 to 1 for the hydrogen peroxide to oxygen. So we can actually see this gas bubbling out of solution here. So you're going to propose some hypotheses again. You're going to propose a null hypothesis, an alternative hypothesis, and as far as forget about this. So I read the procedure and it looks like it's going to take a really long time. So we're not going to reuse the test tubes, we're going to get brand new ones, hooray. And the meat extract I will make myself. I will do that. So we're also not going to quantify the gas production here, and so in part 12, table 11.4 on page 119, that will be required, so that is your results. And so you're just going to be doing the qualitative. So what you're going to do is you're going to look at the bubbles formed. And so create your arbitrary scale of like 0 to 5 for lots of bubbles or no bubbles. However you want to do that, just make sure you tell me what your scale means. Answer all of question 2. Again, I will have graph paper for you. Enzymes sometimes, depending on the enzyme itself, can be inhibited by chemicals in several different ways. So inhibited means that they no longer can function as they need to. They might lose some of their functionality, they might lose all of their functionality, but in some way they are inhibited. So a peroxidase, peroxidase is a plant and bacterial enzyme that acts similarly to catalase. So it also breaks down hydrogen peroxide into water and oxygen. So peroxidase can actually undergo competitive inhibition. So the molecules, competitive inhibition means that the molecules are actually structurally similar to the substrate that binds to the active site. So what it does is that it competes. So let's say we have our Pac-Man enzyme, right? And we have now this substrate that fits like this. Let's say it's a long triangle, and this is our inhibitor. And here is our normal substrate. They are small triangles. Now, if Pac-Man has his face stuffed with pie, as it looks like here, now our little substrates can't get in. They can't bind. So Pac-Man is gorging on pie, and he can now no longer eat Doritos. So hydroxylamine is HONH2, is similar to H2O2 in shape, so kind of like this giant piece of pie, and competes with peroxidase. So that means it won't produce any oxygen. So we're going to measure the production of oxygen with a compound called guaiacol. Now guaiacol will turn from colorless to brown when it's oxidized. So oxidized literally means addition of oxygen. So once we add the oxygen, we should see clear to brown. So the amount of brown color is proportional to the amount of oxygen formed by the reaction. So if you have lots of oxygen, you will have lots of brown color. And vice versa. If you have no oxygen, you will have no brown color. So here is the reaction for that. O2 plus guaiacol, which is colorless, when those combine to form oxidized guaiacol, we will have a brown color. So in procedure 11.3, I'm going to make a turnip extract. So I hope I can actually find some turnips. Otherwise, I'll have to find an alternative source of the peroxidase. So we're going to be using the spectrophotometer for this. So I will give you a rundown about what the spectrophotometer does and how to use it after we start our first experiment. So if you look at the table on on page 121, table 11.5, should be a dot there. That's all right. Table 11.5, it is very detailed. So the written instructions on page 120 actually have the written out instructions, but I really highly recommend you use the table. Put your tubes in order, add water to all of them first, then your guaiacol to the ones you need, then hydrogen peroxide, then turnip extract, then the hydroxylamine. Okay, I hope I can find some hydroxylamine so we can actually do this experiment. So you will also you can complete the table 11.6, because you're only going to be doing this for about 5 minutes, which is nice, and you'll use the spectrophotometer to figure that out, the absorbance. So you're going to answer question 3, all parts including graph. Again, I will have lots of graph paper for everyone, and the questions for further thought and study, numbers 3.5. I hope this has been a pretty good numbers 3 through 5. Sorry, 3, 4, and 5. So I hope this has been a good introduction to this. It's not much longer than the last one, which is good. And I will see you all tomorrow on Thursday.